Hello, everyone. Our body is home to trillions of microorganisms, including bacteria, fungi, viruses, and archaea. Together, these microorganisms form what is known as the human microbiota, while their collective genetic material is called the microbiome. Most of these microbes are not harmful. In fact, they play essential roles in digestion, vitamin synthesis, immune regulation, protection against harmful pathogens, and even drug metabolism. But what happens when this delicate balance is disturbed? This condition is known as dysbiosis. In today's lecture, we'll understand what dysbiosis is, what causes it, how it affects health, and the current approaches to restoring a healthy microbiome. What is dysbiosis? The term dysbiosis simply means an imbalance in the normal microbial community that lives in or on our body. Although dysbiosis can occur in different parts of the body, such as the gut, skin, oral cavity, or vagina, the most widely studied form is gut dysbiosis. Under normal conditions, beneficial and potentially harmful microorganisms exist in a balanced ecosystem. This balance allows the microbiota to support digestion, maintain the intestinal barrier, regulate the immune system, and prevent the growth of disease-causing microbes. Dysbiosis occurs when this balance is disrupted. This disruption may involve one or more of the following changes: a reduction in microbial diversity, meaning fewer different types of beneficial microorganisms are present; an overgrowth of harmful or opportunistic microorganisms that normally exist in small numbers; or the complete loss of beneficial bacterial species, reducing the microbiota's ability to perform important physiological functions. When these changes occur, the normal relationship between the host and its microbiota is disturbed, which may affect overall health. Now, what causes dysbiosis? Several factors can alter the composition of the gut microbiota. One of the most common causes is antibiotic use. While antibiotics eliminate disease-causing bacteria, they can also destroy beneficial microorganisms. Even a single course of broad-spectrum antibiotics may alter the gut microbiota for weeks or even months. Diet also plays a major role. A diet rich in dietary fiber, fruits, vegetables, and whole grains supports beneficial bacteria. In contrast, diets high in processed foods, refined sugars, and saturated fats may reduce microbial diversity and promote the growth of less beneficial organisms. Other contributing factors include chronic psychological stress, which can influence the gut through the gut-brain axis; gastrointestinal infections, which temporarily or permanently alter microbial populations; excessive alcohol consumption, which can damage the intestinal barrier and disrupt microbial balance; certain medications, particularly proton pump inhibitors, commonly called PPIs, and non-steroidal anti-inflammatory drugs, or NSAIDs, have also been associated with changes in the gut microbiota. Finally, intestinal diseases, gastrointestinal surgery, and other medical conditions may significantly alter the microbial ecosystem. Researchers have linked gut dysbiosis with several diseases. One of the strongest associations is with gastrointestinal disorders, particularly irritable bowel syndrome, or IBS, and inflammatory bowel disease, known as IBD. Dysbiosis has also been associated with obesity, metabolic syndrome, and type 2 diabetes mellitus, suggesting that gut microbes influence energy metabolism and glucose regulation. Changes in the gut microbiota have also been linked to allergies and asthma, where alterations in immune regulation may play a role. In addition, researchers are investigating the relationship between dysbiosis and autoimmune diseases, including rheumatoid arthritis and multiple sclerosis. Another fascinating area of research involves the gut-brain axis. Communication between the gut microbiota and the brain occurs through neural, hormonal, and immune pathways. Because of this connection, dysbiosis has been associated with conditions such as anxiety, depression, and other mood disorders. However, it is important to remember that in many of these conditions, research is still ongoing. Scientists are still trying to determine whether dysbiosis is the cause of disease, a consequence of disease, or simply one of several contributing factors. Can dysbiosis be corrected? The good news is that the gut microbiota is dynamic, meaning it can change over time. One of the most effective ways to support a healthy microbiome is through diet. Increasing the intake of dietary fiber and a wide variety of plant-based foods provides nutrients that beneficial bacteria use for growth. Fermented foods, such as yogurt, kefir, kimchi, and sauerkraut, may also support microbial diversity. Probiotics, which are live beneficial microorganisms, have shown benefits in certain medical conditions. However, their effectiveness depends on the specific bacterial strain, the dose, and the condition being treated. Therefore, probiotics should not be considered a universal solution. Another important strategy is the judicious use of antibiotics. Antibiotics should only be used when medically necessary to minimize unnecessary disruption of the gut microbiota. Finally, maintaining a healthy lifestyle through regular physical activity, adequate sleep, and effective stress management also contributes to a healthier microbial community. As our understanding of the human microbiome continues to grow, maintaining a healthy gut microbiota through a balanced diet, responsible antibiotic use, and healthy lifestyle practices may become an increasingly important component of disease prevention and personalized healthcare. Thank you for watching. If you found this lecture helpful, don't forget to like, share, and subscribe for more educational content on pharmacology.